Maybe we can get started. Hello, everyone. Thank you for joining us for the Statistical and AI Methods for Health Data Science Seminar Series. This series is organized by the StatSub AI group. So the group aims to organize activities that empower statisticians to participate more actively in AI research and leadership. So recently, the StatSub AI has been officially recognized as an ASA interest group, and then we had the first business meeting during the past JSM. And here's a website, so please sign up for the group and then receive recent news and then seminar series. And then we also have the recordings available at the YouTube channel of the group. Here's the steering committee and then also the current inaugural officers. Today, it is my great pleasure to introduce Dr. Taki Shinohara, who's a professor at UPenn. He is also the director of the Penn State's Imaging and Visualization Endeavor, the PennSIV. Taki also plays a lot of central role in interdisciplinary centers at Penn, including the Center for Clinical Epidemiology and then Biostatistics and the Institute of Biomedical Informatics. Taki's research focused on the structural and functional brain changes using quantitative and biomedical imaging, particularly in neurological, psychiatric, and developmental disorders. And then recently, Taki's group has made a lot of contribution in terms of imaging data harmonization, which is also the topic of today's seminar. And Taki also received several honors, including also he has been elected as the fellow of the ESA. Without further ado, so let me hand it to Taki, and then also here are some logistics as well. So please, if you have any question, please type it in the chat, and then I will monitor the questions and then ask Taki to pause maybe, and then answer your question. And then if you have for example more generic or general questions, please save it to the end. We will have some Q&A time at the end, and then without further ado, just Taki, please take it over.
Thanks so much, Yi, and thanks very much to Andrew and Papan for the invitation to be a part of this seminar series. I'm very excited to hear about this effort and look forward to being a part of it. Okay, hoping everyone can see my slides.
Yes.
Perfect. These are my disclosures. I want to start out today by just mentioning some of the folks who are responsible for the work that I'm going to describe to you. I'll be talking about some developments that our group has done, as well as many collaborative efforts and work by others in the field. And I'll point out particular contributions as we go, but many thanks to the people who actually did this work. Let's try this again. And next, I want to tell you a little bit about a couple of motivating examples. I understand there might be some folks here who are neuroimagers through and through, and there might be those who are more general statisticians or AI experts. And so I figured I'd just give a couple of examples of the kinds of reasons or kinds of studies we might do with neuroimaging and how quantitation and how AI is important in these kinds of problems. So one problem is near and dear to my heart. That's the problem of multiple sclerosis lesion detection and study. This is something we've worked on for some time. For those who may be less familiar with this particular problem, multiple sclerosis is an immune-mediated disease of the central nervous system. It's cross-sectional problem. This is about 1 in 750 to 500 Americans. And the hallmark of multiple sclerosis is the inflammatory demyelinating lesion that occurs in the brain and in the spinal cord. And what you can see here on the right, this is a particular kind of MRI. It's called the T2 weighted FLAIR image. This is a single slice acquired, or I should say shown here in an axial view. So this is parallel to the ground. At the end of this giant blue arrow, in case anyone can't see, there is indeed a hyperintensity or an area of brightness that's occurring just next to the ventricles. And these lesions, they form in the brain of people who have MS. In both the white and gray matter, they're more easily visualized in the white matter, and their accumulation is known to be associated with morbidity and disability, and it's thus monitored, or should say these lesions are thus monitored in the clinical management of multiple sclerosis. They're also key factors in the diagnosis of multiple sclerosis, and they're used in quantitative forms, both now increasingly in the context of clinical care, but also in clinical trials for novel therapeutics in multiple sclerosis. Another area of research in which neuroimaging, quantitative neuroimaging has had great impact outside of neurology, where multiple sclerosis sort of sits, is in psychiatry. And this is a figure that comes from Machen Yu's work. This is a collaborator who is now in Indiana. At the time was actually here at Penn as a postdoc with Yvette Chalene, who is very interested in heterogeneity in depression. So unfortunately, first-line antidepressants are rarely, or should I say, are not always effective and not often effective in people who present with major depressive disorder. And the heterogeneity in terms of disease has been considered and is studied often in the context of neuroimaging to be able to better parse expected therapeutic response. And so this is some work demonstrating the kinds of connections between brain networks that we see in functional using functional MRI that depict the kinds of changes we see in major depressive disorder. So in many different areas of medicine, we see imaging being incredibly important in the context of diagnosis, care, and research. But these are just two examples to sort of hopefully motivate you that there are indeed there's clinical impact and basic science knowledge to be gained from using these techniques. So next, I wanted to give you just a very brief introduction to biomedical image analysis. So when I talk about imaging, I talk about images very differently from the way that my parents talk about them. My parents are actually in the field of, or in the humanities. My mom's basically an art historian, and she thinks about images in the context of religious figures or historical moments and things like emotion and color and so on. For me, as a statistician, images are data, and we view images as arrays. This is an example of such an image. This is actually, again, someone who has multiple sclerosis. This is a T2 weighted, I should say T1 weighted post-contrast image. But in any case, you can see that it looks like a picture here on the left, but if we zoom in this red little box here, I think this figure actually came from Elizabeth Sweeney, so many thanks to her for providing this many years ago. In each box here, or so-called pixel, we have a number. And so when we think about images, we can think of them just simply as arrays. And these arrays can be very large because images are acquired at all different kinds of resolutions. Sometimes we have hundreds of thousands or millions of measurements, or tens or hundreds of millions of measurements, depending on the particular imaging modality. How do we analyze images? Well, the field of image analysis in neuroscience and so on really basically takes a three-step approach. The first is to acquire the images using some kind of a scanner, be it an MRI scanner, a CT scanner, a PET scanner, whatever tool allows you to be able to visualize the tissue you're interested in. And second, those images go into an image processing algorithm that tries to remove artifacts and extract features of interest, which then go into a table. And this is a simple example of a table in which there are perhaps 8 features being measured and well, I guess 6 subjects or six lesions, whatever they are, being analyzed here. If you're interested in learning more about image processing, I definitely suggest you check out the Neuroconductor platform. There's a nice paper in Biostatistics from a little while ago describing the work by some folks at Hopkins that really sort of pushed forward this initiative to be able to process images in R, a language that we're all very familiar with as statisticians. So one might ask, okay, is that the story? Once we have our tabular data, are we done? We can analyze images from observational studies. We can analyze images or features coming from images in randomized controlled trials. We can use images as predictors or outcomes. We can do all different kinds of things, ranging from simple t-tests to advanced deep learning. Are we good? Do we need to think about the fact that this table came from images? And I would like to argue that indeed we do, because images have their own complexity in the context of acquisition, in the context of processing, that really doesn't matter if we're going to try to draw biological knowledge from those data. And in particular, I'd like to talk to you today about the problem of interscanner biases in biomedical imaging studies. So we've known over the past 20 years in the MR physics literature, in neuroimaging literature, image processing literature, that if you take images on different scanners and extract features from those scanners, you get different answers. Indeed, also, if you process the images differently, you get different answers. And these are just some examples of what happens in the context of morphometrics. So we're talking about as something as simple as you put a participant in the scanner, you measure the size of their brain, and you do it again that same hour on a scanner next door, and you get slightly different measurements, sometimes more than slightly different measurements in the context of simple volumetric assessments. And again, this literature dates back 20 years. There's maybe not so much new here, but it's certainly something to keep in mind as people consider comparing, for example, differences in reported average brain size and disease and so on across different groups and different centers. But increasingly over the past, 15, 20 years, neuroimaging has really shifted from single center studies of I have my participants I've recruited here and I'm going to image them to large multi-center studies. And these include, for example, the Alzheimer's Disease Neuroimaging Initiative. The IDNI consists of thousands of people who either have Alzheimer's disease, have mild cognitive impairment, or are healthy controls. The ABCD is tens of thousands of kids being studied because they're to better understand neurodevelopment and risk for addiction. And the UK Biobank, which is 100,000 people just living in the UK. And to be able to achieve these kinds of sample sizes, it's unfortunately no longer feasible to use one scanner at one center to image everyone. Indeed, this is a sort of a cartoon drawing of a simple study that has 3 sites. And at these 3 sites, different scanners are used. So at site 1, I should say, scanner A is used for a while, and perhaps scanner A eventually is taken offline, and scanner B is used after scanner A is decommissioned. At site 2, there are 2 scanners, and perhaps site 2 is a very large research site. So these are 2 research dedicated magnets, but because there's so many scans going on, when a participant comes in, they'll either be scanned on scanner D or scanner E. And if they're coming in over time, they'll be scanned, either on either scanner, just based on when the scanner is available and when the participant's able to come in for a visit. And here you can see scanner 3, or I should say site 3 just has 1 scanner. Now, we're not talking about just 3 scanners, or I should say 3 sites and 5 scanners in many of the studies I just described to you. It's many more than that, but I just wanted to give you an idea of the problem. So one might ask, well, we know that there are differences across scanners because we have this literature back in over the past 20 years in terms of volumetric differences. There are also literatures in terms of functional MRI and diffusion MRI and all different kinds of assessments. But how much does it really matter in terms of studying disease? Well, in the next few minutes, I'd like to try to convince you that it can matter a great deal. So what you can see here is a study protocol that was developed by a group called the NAMES, the North American Imaging and Multiple Sclerosis Cooperative. And this is a consortium that we're a part of that's now, I think, 40 sites across the US and Canada. And but it started, this project started about 15 years ago, when a group of 7 sites wanted to get together to be able to share data, acquire data together in research studies. They had different scanners. You can see here, these are all scanners made by Siemens and acquiring data at a specific field strength. So some of the technical features of the scanners were similar to make the problem a little bit easier. And these, you don't need to read all the details, but basically the idea is that there were a couple of different images. Actually, there was a sequence of different kinds of images that were taken, and all the protocols were matched very carefully to provide as similar images as possible. And when we had a meeting to discuss this protocol and decide on these protocol sort of features so as to be able to make the scan as similar as possible across these 7 sites, one of our collaborators in the group asked, well, how do we know if we did well at the end of choosing all of these numbers, these parameters for optimizing how to make the images similar? And to be able to assess that, Danny Reich's group at NIH recruited a participant, a 45-year-old man with stable relapsing remitting MS, who actually traveled to each of the sites after we set up this protocol during a period of 4 months. This gentleman was radiologically and clinically entirely stable, and traveled to these sites so we could image him and just be able to sort of check that the harmonization was successful. And while we have studied this one participant in many different ways, these images from this one participant in many different ways, I'd like to focus for the near time future just on a simple assessment, that is the volume of those white matter lesions that I mentioned to you in the earlier example, that we know to be associated with disability and morbidity, and indeed are also used in the context of therapeutic decisions in the care for people who have MS. So someone who has a new lesion or a set of new lesions will often be considered to require additional therapy or a change in dose of therapy or so on. And this is the result from that traveling subject study. In columns here, you can see acquisitions taken at 4 different sites. In the first, we have a scan on, this is the actual model of the scanner, a Skyra, a Siemens Skyra at the Brigham up in Boston. In the second column, you have again a Skyra at the National Institutes of Health in Bethesda. In the third column, we have a Trio, a previous generation scanner at Oregon Health and Sciences University, and then a Vario out in LA. And first, you can see that these images, these are T2 weighted FLAIR images, and there are red manual delineations of the boundaries of lesions so that we can measure the volume in the most precise way at the time. And these, first of all, I should say these are sagittal acquisitions. You can see taken images taken sagittally and or visualized sagittally. And in yellow, you can see boxes in the first row that are actually enlarged in the second row. And if we take a look at the images acquired across scanner, you can see in these enlargements that there are indeed several differences. Perhaps you can see that the previous generation scanners, the Trio and Varios, actually seem to be more grainy in terms of their image quality. And furthermore, if we take a look at this lesion that's taken, that's shown in the yellow arrow in the top row, actually at the Brigham, it looks twice as large, volumetrically twice the size as it does on the OHSU scanner. And you can see here in the bottom row that there is what was annotated as a lesion on the Skyra at NIH that's actually not visible at all at OHSU and looks like a vessel on the Cedars-Sinai and it's barely visible on the Brigham scan as well. So this picture immediately sort of set off alarm bells in our mind because we thought, wow, this is the same participant, biologically totally stable, but technically we see variation, both in terms of huge differences in terms of volume and even the appearance or lack of visibility of lesions. And here you can see this quantified on the y-axis here in blue. You can see that those T2 weighted lesion volumes, I won't talk about the red dots. These are T1 hypointense measurements, but if you're interested, I can tell you a little bit more about that. In the first and third columns, you can see measurements taken at the beginning of the study, that 4-month study period, and at the end of the study, indicating that the lesion volume was indeed very stable across acquisitions at the NIH. But across scanners, we see pretty remarkable differences. And indeed, we also see quite a shift from the Skyra scanners to the previous generation Trio and Vario scanners. And the magnitude of these differences can be up to 4 milliliters, which is up to a quarter of the total volume of lesion in this person who's been living with MS for decades. So quite shocked to see the magnitude of these sizes, magnitude of these differences. One might ask, maybe that's just something about the way we measure lesion volumes. Well, the thalamus is a gray matter structure that's one of the earliest and most severely affected by atrophy, loss of brain tissue in multiple sclerosis. And you can see here 4 different techniques we use to be able to measure the volume of thalamus in this same participant across scanners. And it's quite remarkable to see that the scanner explained a great deal of variation, and there were really prominent systematic differences across the acquisitions. So I hope I've convinced you that even in one person with MS, the variation that we can see in terms of differences across scanners after harmonizing the imaging protocol by trying to tweak the protocol to make the images similar and using only Siemens 3 Tesla instruments of similar magnetic resonance imaging scanners can be quite large and could certainly drive differences in inference. So the question then becomes, what can we do to address this? How can we fix this? Well, as a group of statisticians who might say the simplest thing to do to deal with mean differences across different scanners is to treat scanner like any other nuisance variable that we have. We could just adjust for scanner or site in a regression model. And that, of course, helps a great deal. Other groups have also proposed different kinds of techniques for addressing these differences in scanners. This is some work by Roland Henry's group at UCSF, led by Nisha Shinohara at the time. And in that paper, they described a scaling approach that seemed to help quite a bit with respect to making measurements similar. But these methods don't consider a key factor, which is that when we assess the brain, we don't usually just take one measurement, we usually take many measurements. And if we do corrections one at a time, that could really be suboptimal when we know that the systematic differences that we are observing across scanners actually are shared across features to some extent. So to talk more about that, I'd like to describe another study to you, maybe another story to you. This is some work that was done by Jean-Philippe Fortin, who was a postdoc in our group and just joined us from Johns Hopkins at the time, in a collaboration with Ragini Verma, who's an applied mathematician and diffusion tensor imaging specialist here in our Department of Radiology. And Ragini was very interested in combining data across different studies in which diffusion tensor imaging was acquired. And in particular, she wanted us to sort of collaborate on a project where images were acquired at two particular centers with two particular imaging protocols. And to work together, what we decided would be helpful would be a carefully controlled set where we have 105 participants of a particular age range, and biological sex distribution imaged on one scanner and one DTI protocol, and then 105 imaged on the other scanner and the other protocol. And the idea here was basically to develop tools or figure out how we could do something to the data so as to be able to make them more comparable and jointly analyzable. So the first thing that JP did when he received these processed images from Ragini's group was to basically do the process I described to you earlier, take these images and turn them into a matrix that consists of different scans by different locations in the brain. I should say that these images have all been registered, so the same location corresponds roughly to the same location in the brain. And he did this for two different kinds of DTI maps. I'll tell you a little bit more about them in a few minutes, but fractional anisotropy and mean diffusivity maps. And again, the goal was to be able to take data from these two studies and conduct some kind of a procedure on them so that the data set could be combined. Well, as a good data scientist, the first thing that JP did was took a look at his data. So here you can see each line corresponds to a histogram, a density smooth from a histogram from a single participant. And on the left, we have fractional anisotropy. On the right, we have mean diffusivity. These are just two different ways of being able to measure the integrity of the white matter. And you can see in colors the sites. And it's pretty clear, especially in fractional anisotropy, that there's a great deal of difference across these two sites. And remember, these participants are age and sex matched, so we would expect the measurements to be quite similar biologically, but in fact, what we observe is quite different. In terms of mean diffusivity, we also see that as well. The next thing JP did was he calculated what he called MA plots, I think many of us also know as Bland-Altman plots. So here on the left, again for FA, and the right for MD, each point corresponds to a particular location in the brain, a particular voxel in the white matter. And on the horizontal axis, we have the average of the two measurements across site 1 and site 2. And on the vertical axis, we have the difference between those two averages. And of course, we expect to see a flat line at 0 if there are no systematic differences across sites, but we see something far from that. Indeed, also, we seem to see some sort of substructure in these measurements, I should say, in these patterns across measurements. And indeed, if you take a look at this spiky region up here in this blue ellipse, that actually turns out to be regions of the posterior in the back of the brain. So there seems to be spatial information, or I should say a spatial relationship with these deviations, with these scanner effects. So then JP was very interested in this problem because he had had a history in the field of batch effect correction. He'd done his PhD in the field of batch effect correction for gene expression studies and thought, well, maybe we can apply some of the tools that are out there for genomics and to be able to solve this problem and mitigate these differences we see. And here are some examples of techniques that he had previously developed. And then, of course, some of the most commonly used approaches in the gene expression literature. And what he found worked very well was a technique called combat. So combat, as many of you, I'm sure, are aware, is a paper published for gene expression batch effect correction by Johnson, Lee, and Rabinovich in Biostatistics in 2007, so a decade before. And it's really just a mixed effects model with a little bit of extra spice on top. So the model is that the measurement for site I, it's in sample J or subject J at location or voxel V, can be broken down into two parts. The first is a part relating the image intensity of the value of that image at that particular location for a participant with a certain pattern of or certain number of covariates that are to be adjusted for, that are to be sort of preserved in the harmonization. You can think demographics as well as biological, I should say disease-specific covariates, things you don't want to remove in a harmonization. And then there's this term for, I should say, a location term that explains differences in means across scanners for every location. And importantly, there's also this scaling. This scaling factor on the residual adjusts the amount of noise in the measurements. And this is key because as you saw from those pictures I showed you from the NAMES pilot experiment, there were actually differences in smoothness that propagate into differences in noise, in volumetric and other measurements. So to be able to adjust for differences in those variances is also key. And the combat method, as proposed again by Johnson, Lee, and Rabinovich, can be fit using an empirical Bayes framework by specifying some priors and estimating the hyperparameters from those data, and that can be solved iteratively. The procedure can be solved for iteratively in this very simple formula. And this is an example of such a correction. Here you can see that there definitely is a difference across sites in terms of this mean effect. But fascinatingly, we also see that the differences across, I should say the mean effects, the differences across sites in mean, they seem to also track biology insofar as they track well-established sort of tracts in the white matter of the brain. And this is fascinating to us from a technical perspective, but also terrifying to us from sort of an epidemiologic perspective, because one could very easily imagine that they would do a study across a couple of sites in which there's imbalance in terms of some factor of interest, and then do an association study without addressing the interscanner differences, and find a map like this and make a biological story about it, when in fact, this could simply be described, or I should say, could simply be explained by heterogeneity in terms of the scanners used or differences across sites. So here are those Blaine-Altman plots again. I showed you in the top left, there's the raw measurements that I showed you before. And here are a couple of different ways one could consider harmonization. Scaling, this is a functional normalization. These are two techniques that Ravel is an image normalization method, and SVA is another batch effect correction tool that's been proposed and is very useful in the gene expression field. And you can see very nicely that SVA and combat both result in a mitigation of this side effect. This cloud goes right down to zero at this horizontal line. I should say zero horizontal line, which is very nice. And this similar sort of phenomena happens in the context of MD. Now, in terms of what this helps us to learn, well, first, if we take a look at the number of voxels associated with site in FA pre-harmonization, basically the entire white matter shows a difference, but after harmonization, there are no such differences. With MD, we see a similar effect. And indeed, post-harmonization, whereas we didn't see much in terms of FA effects, FA differences associated with age in this neurodevelopmental sample, we actually do see some after harmonization with combat. And similarly, with respect to MD, we see an increase in the number of features that are associated with the phenotype of interest age after harmonization. This is actually something that's sometimes challenging for us to explain to some colleagues and collaborators that we don't always expect to see this, right? If you have a study in which there's a null effect, but you have a strong side effect, having said that, the phenotype of interest is imbalanced across sites. It's a good thing if you see less signal because it's not real signal, it's noise, right? But in this particular study, we were excited to see that actually mitigating the interscanner difference using these harmonization techniques actually facilitated a reduction in the noise and unveiled additional neurodevelopmental associations. We also, I should say, JP also did a set of analyses using concordance of the top plots based on simulations in which he induced imbalance associated with a particular feature of interest by resampling. And there he found combat to perform much better than SVA simply because combat facilitates the adjustment by those features of the X beta, the covariates of interest that are to be protected for, whereas SVA doesn't have an option for that, doesn't model that. JP then did another study looking at, instead of DTI measurements, these are volumetric measurements, in particular cortical thickness assessments, in a 4-site study of depression, that same study that I mentioned to you that Mei-Chen worked on using fMRI. And this is a collaboration with Yvette Chalene, in which he found that combat actually quite nicely mitigated interscanner differences and facilitated more rigorous modeling by addressing intersite differences in cortical thickness as well. In that same study, this is work again by Mei-Chen, and this is a collaboration with Kristen Lin and Yvette Chalene, in which Mei-Chen looked at those functional data I was describing to you and found that actually if the goal is to be able to mitigate interscanner differences in terms of connectivity between predefined regions of interest, these are connections in the brain that we can measure, or should say functional associations of the brain that we interpret as functional connections in the brain. We can use combat as well, or adapt combat only slightly so as to be able to mitigate those interscanner differences. So one might ask, okay, we have this tool that came from the gene expression literature, and it's been cited extensively and proven extensively in a wide variety of settings in that world, and now we see that folks across the world are also using it for things like PET imaging and PET MRI and CT imaging and so on. Is the problem solved? Do we need to do more methodological work, or are we happy with simply adapting or borrowing this tool that was developed in gene expression literature for the problem of interscanner harmonization in MRI? Well, I would argue that the problem is not solved, and the reasons why are several-fold. But the two key sort of motivating sort of directions for our research in this area have been the fact that in neuroimaging studies, we are interested in different things and thus use different study designs to be able to study those associations that, or I should say the biology that differs quite a bit from what we're interested in gene expression studies. And similarly, to be able to study complex signals in neuroimaging, we use quite different analytical tools, which again require different kinds of harmonization. So to give you an example in terms of study design, this is some work done by Joanne Beer, who's a postdoc with Kristen Lin and myself, again, working with Yvette Chalene, in which she asked the question of whether harmonization using combat was sufficient in longitudinal neuroimaging studies. So when people are scanned perhaps at one site on a couple of different scanners, but then there's a switch out of one of the scanners, or at another site where a scanner is lost. And so people are flip-flopping between scanners, which we saw from that NIMS pilot study can result in rather remarkable differences. How can we address those interscanner differences? And what Joanne found was that using classical cross-sectional combat harmonization actually doesn't address the problem insofar as residual side effects exist, and in fact, new side effects can be introduced if methods aren't appropriately applied. And furthermore, she proposed a new combat-like tool called longitudinal combat that facilitates harmonization in these kinds of settings. Another question might be, what happens if we need more than just mean variance correction? So in the combat model, which I've drawn again down here, so here that A plus X beta, that's the stuff that I was talking about in terms of protecting variables of interest that may differ across site, but that we'd like not to harmonize out. And this is where we found combat to be more successful than SVA in those experiments that I was describing to you. Well, the model is basically just that there's an additive effect of site across our scanner, and then there's this multiplicative, marginal multiplicative error term. In fact, in our experience, side effects are much more complex than this model would explain. Now, if the goal is to just do marginal association studies, so-called voxel-based analyses or feature-based analyses, combat can be, very, very helpful if we're just going to do a t-test at every location of the brain, for example, or regression at every location of the brain, or in the context of longitudinal combat, a mixed effects model at every location in the brain. But if our goal is to do something more complex, like take a set of features from across the brain and incorporate them using some kind of multivariate statistical or machine learning model to be able to study associations with phenotype, it turns out that combat also fails to address the covariant, the complex side effects in the covariance structure. This is some work by Andrew Chen, who's now at MUSC, and Hashang Shou, another colleague of mine here at Penn, in which Andrew demonstrated that while combat can be helpful for univariate analyses, using combat on multivariate data and then applying multivariate pattern analysis actually doesn't mitigate the scanner effects sufficiently, and residual effects remain. So this is sort of an example here. These are some data from the ADNI in which we have 3 different sites here, and this is the average cortical thickness covariance structure on these 3 different scanners, 1 by Siemens, that same manufacturer I was describing to you earlier, and 2 by another manufacturer, or 2 sites that use scanners from a different manufacturer, General Electric. And you can see that the covariance structure across measurements is very different. And if you combat adjust the data and then again take the covariance, look at the covariance structure, again, you see that the covariance structure is very different because we aren't actually changing the correlation structure. But Andrew developed a technique called CoveBat that I'll describe to you on the next slide that actually addresses differences in the mean, the variance, and the covariance. And the CoveBat model is a very simple model. It basically assumes a low rank structure on the error terms and on the across features this is now. And also that it assumes that the side effects in this in the covariance can be expressed in that lower dimensional space, which facilitates a simple, well, 4-step approach. First, we residualize out mean and variance effects. And then we basically apply combat in the PCA space, which allows us to be able to come up with a correction and then a reconstruction of data that are indeed harmonized insofar as the side effects in mean, variance, and then covariance are addressed. And so here are some results from that work published in Human Brain Mapping almost 5 years ago now. Here's the AUC for detecting whether an image was acquired using a Siemens scanner. It's 0.9 basically on unharmonized data. It goes down to 0.6 something with combat, but actually a random forest, I should have mentioned, this is a cross-validated random forest in these data, actually can only detect or really can't detect whether a scanner is whether an image is acquired on a Siemens scanner or not. The AUC is about a half. Indeed, also here, we were delighted to see that post-harmonization, we saw an improvement in terms of the random forest facility to be able to determine whether a participant had Alzheimer's disease or not, and also detecting biological sex. So it seemed this technique seemed to, in this particular data set, very much help with unveiling biological effects and mitigating technical variation. Another direction that we've seen harmonization go in over the past few years, and it's really been sort of a very quickly growing field, is in the deep learning sphere. So this is actually from a review that Fengling Hu, who's an MD PhD student in our group, just finished his PhD and is now back in med school. He wrote in Neuroimaging 2023, and he described in that review, which is now a couple years old, a set of many tools that were, or I should say, the variety of tools that had been at the time proposed for using deep learning to mitigate interscanner differences, many of which tried to be able to disentangle basically stuff that you care about, biology, so-called content, from stuff that we don't, or style in the scanner. And the goal of many of these tools is to be able to find a latent space encoding that's really independent of that scanner, and then to be able to do reconstruction by combining information in that latent space that's sort of biological information with the scanner information, or I should say the scanner ID or scanner manufacturer, to be able to do a reconstruction. A popular approach for this has been the conditional variational autoencoder, the CVAE. And these CVAEs, again, try to learn a latent space distribution for each subject and use loss function to be able to incorporate reconstruction and facilitate this encoding. But these CVAE approaches, I should say, actually have some pretty strong assumptions. So, for example, one of the assumptions is that an encoder can find a latent space that's truly independent of scanner. Another assumption is that a decoder can perfectly reconstruct the data. And finally, these techniques to date haven't been able to, or I should say, at that time, up until that date, I hadn't been able to deal with additional covariates. That same problem I was describing to you in terms of the SVA method, for example. And so these limitations, we believe, have hindered their adoption and use. Because, in fact, generally, in our experience, scanner effects can be minimized but not eliminated. We can fit data very well using deep learning, but we can't really fit data perfectly. And we often have imbalance in covariates. For example, there are differences in demographics across particular acquisition locations, which result in imbalance in covariates. So Feng has been working on deep learning approaches and studying the effects of deep learning harmonization methods in the context of a couple of real world examples. And in this particular study, this is work he did in an area, or I should say, to develop a method called deep combat. What he found was that some of the tools that were out there, for example, some of these conditional variational autoencoder methods, really changed the structure of the data a great deal. So these are PCA plots showing the data raw, the data post-combat. Color here corresponds to, again, acquisition manufacturer, whether the scanner was taken on, whether the image was taken on a Siemens scanner or not. And in the bottom here, you can see what happens after harmonization using these variational autoencoders. And the data really look nothing like they did before. So what Feng proposed, instead of using these variational autoencoders alone, was to incorporate them into a hybrid statistical deep learning model, both to use an autoencoder sort of architecture, but then also to incorporate residuals and corrections of those residuals in the context of side effects, preserving biological associations using that regression in terms of to be able to reconstruct and then basically harmonize or synthesize what data would look like had they been acquired on a different scanner. And here are some figures that sort of describe that or demonstrate that. Again, here we have PCA plots. Here we have 3 manufacturers in the top left of this panel B, raw, combat adjusted, combat adjusted, and then deep combat adjusted, where we see a little better, a better correction. And you can see U maps in the bottom left. And these are comparisons between harmonized and raw data. Compared to other deep learning methods, these, deep combat, you can see very clearly preserves the shape and the structure of the data much more than these other variational encoder methods did. You can see, especially in UMAP, there's a lot of induced structure from the harmonization, which we think is concerning for our study of biological variation in the data. Furthermore, deep combat seemed to reduce batch effects quite clearly, quite effectively, I should say. These are different machine learning techniques that were employed to be able to detect which scanner was used to be able to acquire an image. while pre-harmonization you can see quite clearly, quite easily which type of scanner was used, post-harmonization we're much closer to chance. And we also see biological associations relatively being preserved in these studies, with sort of more flat differences comparing the raw versus the harmonized data. So I hope from this presentation, you're all terrified of side effects and scanner effects, because we believe that they're strong and they can both obscure associations as well as cause fictitious findings. And indeed, the differences across scanners and protocols can appear in the mean of measurements, in the variance of measurements, in the covariance structure, and even in more complex distributional features of both structural and functional MRI data. And furthermore, connectivity studies pose unique challenges for harmonization, given the level of complexity of these data, and there's still really a lot to be done in this field. So thanks again for the invitation, and I'd love to take any questions you might have.
Great. Thank you, Taki. Really great talk. I don't see any question in the chat yet, so maybe I think right now we can take questions. So if you have any question, so please raise your hand and then I will call your name. Yes, Panda.
Hi, Taki. Yeah, very nice talk. Thanks so much for that. I just have a quick question. So, while you are applying either combat or longitudinal combat, do you have any concerns if, like, I have multi-center studies and these, the demographics, I mean, the cognitive, diagnosis and the age are very different from different studies. If we want to harmonize these, cohorts together, is there any takeaway messages that you'd like to share in addition to the the scanner effects and the different parameters or the radiological sequences issues?
Yeah, absolutely. Thanks so much for that question. So we think it's really important to model biological associations during the harmonization during the harmonization process for that very reason. And the combat method is very nice because it has a linear model structure, so as to be able to preserve associations with differences in cognitive status or differences in demographics. But also, it's quite common for more complex associations to exist. So for example, in aging studies or in lifespan studies and early age studies, we often see nonlinear effects, and we can see sometimes more complex interactions in terms of nonlinear effects. And that makes things more challenging. So there's a method called Combat GAM developed by Ray Pompanillo with Hashang Shou and Chris Statistical at Penn that tries to use generalized additive modeling to give more flexibility to an analyst when they're doing harmonization to preserve those associations. But that's really key. In the context of more complex covariance structures in terms of patterns of association with demographics or cognition, that's also a key problem. And I think it's understudied and requires really more methodological development. So there's a lot to be done here.
All right, thank you.
Yeah, I also have a question for Taki. So Taki, thank you for very clear talk and a very comprehensive review. So I'm wondering for those covariance structures, covariance batch effects, how important to really address that? I think so nowadays neuroimaging have such a huge data. So what is scenarios we really need to address that? That's my first question. Yeah.
Thanks so much. This is also a really important question, and it's one that we get oftentimes because I think we're in a little bit of a cottage industry of different extensions of methodologies for minor improvements, and sometimes it's hard to tell exactly what's necessary. But for me, I think going back to the basics in terms of study design and analytical target, that's where sort of we focus. So if your goal is to be able to take some data and do machine learning, our experience has been that adjusting for covariance can be very helpful, because even after adjusting for mean and variance differences in terms of differences between scanners, we can still predict pretty well with accuracy, sometimes in the 70s, 80s of percentages in terms of accuracy, whether an image was acquired on one scanner or another. So if you can imagine that if you have great imbalance in terms of the phenotype you're interested in, in terms of those two sites, those residuals containing that differential covariance structure across sites could be mistaken for having biological variation in that covariance structure as opposed to this technical variation. I don't think I explained that very well, but I guess what I mean to say is we tried to mitigate side effects in whatever structure we're going to try to extract the biological effects from. If it's marginal associations, marginal correction is maybe what we'll do. If it's more complex multivariate structure, then we'll try to do multivariate corrections.
Yeah, so actually, you have already alluded to that. This is my actually following up second question. Is there's a huge imbalance of the covariance across different sites? Do you really think we can correct for the side effects? It's very hard to separate the side effect and the covariance effect. That's my general understanding.
Yeah, absolutely. And this occurs also in the mean and variance effects too, right? If you have near perfect confounding between the scanner and the phenotype of interest, I'm not sure I believe it. So we are often asked by some of our colleagues and collaborators, oh, I have this data set of controls from one scanner from 10 years ago, and now I have this new group that I've imaged on this new scanner using a different protocol. I want to combine them. I don't know how to help you. If that
Thank
makes
you
sense.
very much. Thank you.
Yes, Julia.
See, there I am. Thank you for a wonderful talk. I was just wondering, has there been any work done on utilizing different scan types from the same scanner to boost the information for harmonization?
It's a great question. Yes, I have not seen very much about that, but that's definitely I've heard of people working on similar studies, but I think that's definitely a direction for future studies and methodological development. So you can totally imagine that if you make measurements on the basis of like T1 and flare imaging, if you have a different
Yeah.
T1 but a similar flare, what does that mean? Or if you, yeah. So I feel like there are a lot of different directions to go in there. Also, scans can be similar in terms of some protocol, in terms of some aspects of the protocol, and different in terms of others. So even for a T1, you may have similar contrast, but different resolutions. What does that mean? How could you leverage shared information that you know about in a meaningful way, as opposed to just making each site one sort of different entity?
Thank you. I really appreciate that.
Any other question for Taki? Can I just ask a quick question, so more like a practical side? So do we need to do harmonization every time we change the model a little bit? So for example, you want to add interactions, then I need to rerun the combat again?
So that's a great question. Our current advice is yes, to be most rigorous. Whatever you adjust for in the context of your harmonization model should be what you're going to study and adjust for in the context of the subsequent models. And it's all really a question of which conditional independent structure are you inducing when you do the harmonization. But perhaps future methodology will be able to facilitate this. But that's a common question, a common request we get from collaborators and from grant reviewers is, could you please release the harmonized data? Sure, we can, but only for the particular model that we've applied, if that makes sense.
Great. If there is no more questions, so maybe I share my screen again. So thank you so much for the great talk. And then, so this is just announcement for our next seminar. So which will be September 17th from 12 to 1. So offered by Zhengwu Zhang from UNC. Zhengwu will talk about DTI, diffusion tensor imaging data. And then hope to see you next time again. And then thank you so much again for joining us. And then thank you, Taki, for giving the talk. Yeah. Great.
Thanks again for having me.
Thank you very much. Thank you. Bye-bye.
All right, bye everyone.